So we are picking up from where we ended in our previous lesson. We actually spoke about basic introduction to genetics. So in this lesson, we'll be discussing what genetic crossing is. We'll give a brief introduction to it and then move ahead to solve some questions on dihybrid crossing. So when we talk about genetic crossing, it's basically mating two organisms to study their traits. So you mate organism A and B of the same species, and then you are observing how specific traits of those organisms are passed on to their offspring. That is genetic crossing. And genetic crossing helps us to predict what the traits of their babies might have. So you cross rabbit A, that is white, and rabbit B, that is black. So you are observing to see the offspring. Are they going to be white or black? So that is what genetic crossing is. And when you think about genetic crossing, it's more like solving a puzzle. When it comes to solving a puzzle, you have an idea about what you have to form. So that idea that you have will enable you to guess the moves to make. So when it comes to genetic crossing as well, we have ideas about the traits that the parents have. So based on those traits, we're able to predict the trait that the babies will likely have. That is what genetic crossing is. And genetic crossing can be compared to mixing of paints. Imagine having a red and white paint. If you mix them together, you will get a pink paint. So the red and white are more like the parents, and the pink is like the offspring. So genetic crossing, genes also work in similar ways to create traits. So we have genes from parents, and there are two parents. So when they mix together in specific patterns, they form unique traits in their offspring, just like mixing red and white paints to form a pink one. But why is genetic crossing so important? What do you think? Genetic crossing, why is it so important? So the first one has to do with animal breeding. You have a horse that has admirable traits. So what the breeders normally do is they pick that horse and mate it with another horse with desirable traits. Why? Because they are of the view that the offspring of the mating will lead to they having desirable traits from their parents. So that's how important genetic crossing. So what those people are basically doing is genetic crossing. So it helps them to get best traits from animals. For example, having faster horses or friendlier dogs. And then it's also applied in farming, genetic crossing. So there is a plant that has admirable or desirable traits. For example, it's very resistant to disease or produces a lot of fruits. They can't let that plant just die off one day. They will cross that plant with similar types to produce offspring of that plant. That will also be resistant to disease and produce more fruit. So the next one has to do with medicine. So genetic crossing is so important when it comes to understanding inherited diseases in families. So with genetic crossing, scientists are able to explain why certain children of a couple will inherit inheritable or genetic disorders from their parents, and then other children of the same couple might not inherit that genetic disorder. So genetic crossing is so important. So imagine couple, a man and a woman getting married, and then it is observed that there is an inheritable disease in any one of them, or maybe both of them. So scientists can actually predict the probability of their children having that inheritable disease. That is a classic application of genetic crossing. We have two main types of genetic crossing: monohybrid and dihybrid crosses. Monohybrid and dihybrid. As the name suggests, mono means one. So imagine scientists are actually interested in observing just one trait. So when we study, as scientists study only one trait at a particular time, that is monohybrid crossing. So they pick two rabbits, one white and one black. And then the scientists are only interested in observing how the fair color, that is white or black, is passed on to their children or their offspring. That is monohybrid crossing. They are interested in just one trait at a time. And the opposite is dihybrid crossing. For dihybrid crossing, the scientists are interested in two traits at the same time. For example, they are observing how the fur color, the color of the rabbit, either white or black, and then the tail length, these two traits are passed to their offspring. So when during the crossing they are observing these two traits at the same time, that is dihybrid crosses. So you are going to pick a certain question on dihybrid crosses and see how applicable it is in predicting how the phenotypic ratios are going to be and how offspring are going to inherit certain traits from their parents. So here is the question. So in a guinea pig, black fur is dominant over white fur. And if you remember correctly from our previous lesson, we spoke about a trait represented with alphabets. So if you pick a certain trait, for example, maybe fur color being black or white, if one is dominant, we represent it with the capital letter, and the recessive one is represented with small letter. And for clarity, we also spoke about the fact that the same alphabet is used. So if we decide to use capital B for black, which is dominant, then we must use small b for white. So the B is not because of black starting with B. No. It's a choice. So we could have started with any alphabet. We could have said black W for dominant, then small w for white. So keep that in mind. So in a guinea pig, black, capital B, fur is dominant over white. And short fur is dominant over long fur. So the long is denoted by small s. So a guinea pig heterozygous for both traits. Heterozygote, meaning the traits are not the same. So if you pick fur color, on each homologous chromosome, the traits found on each of them are different. So for example, it has black, capital B, and white, small b. That is why it's called heterozygote for both traits. And then when it comes to fur length also, it is S, capital S for short and small s for long. If it was homozygous for both traits, there should be black, capital B, and capital S. Or small b, small s. But since it is black and white and short and long, that is why I say it is heterozygotes for both traits. So let's continue. So a guinea pig heterozygous for both traits, that is capital B, capital S, small s, is crossed with another guinea pig of the same genotype. So this is what is happening. So two guinea pigs. But before I proceed with the question, I have a question for you, or I have something I want to take note of. Now we are saying the guinea pig has a genotype of capital B, small b, capital S, small s. That is its genotype. So what will be the phenotype of the guinea pig that has this kind of genotype? The guinea pig will be black with short fur. The guinea pig will not be white because black is dominant over white. And the fur will be short, not long, because short is dominant over long. But take note of this. In the genetic makeup of the guinea pig, there is a trait for white fur and another trait for long fur, meaning it can pass on this trait to its offspring. So take note of this. The phenotype is influenced by the genotype. So now let's continue, but let's go a little deeper into a little bit explanation of this genotype and how it's reflected in its phenotype. So let's assume these two chromosomes you see on the screen represent the possible chromosome of the guinea pig we have under discussion. Then on this homologous chromosome, there's a section of it indicated by the vertical rectangle that is responsible for the fair color. So that section, that locus, is responsible for fair color. And for fair color, we have two options, which are the alleles. The first one is an allele for black fair, and the next one is an allele for white fair, represented with small b. So that means for fair color, the genotype will be capital B, small b. So capital B, small b, this section controls only the color of the fair. Now on the same homologous chromosome, there is a section that is responsible for controlling the length of the fair. So for the length of the fair as well, it comes in options or variety. One of them is responsible for short fair and the other one is responsible for long fair, denoted by small s. So that means when it comes to the gene or the genotype for length of fair, it is capital S, small s. So the capital B, small b controls the color of the fair, and the capital S, small s controls the length of the fair. So they are independent, they are on their own. So that is what it is. So with this understanding cleared out, let's go back to our question. So now the first task for us is to use a Punnett square to determine the possible genotype of the offspring. So now if you mate these two guinea pigs, what will be the possible genotypes of the offspring that will be formed? Good. So what are the steps involved in finding the possible genotype? So let's assume these are our two guinea pigs, that the mom, that is male and female. So what do we know about their genotype? As said by the question, their genotype is BBSS, capital B, small b, capital S, small s, for both parents. Now, when we want to mix them using Punnett square to determine their genotype, we should first go ahead and find their gametes. Why is that the case? During sexual reproduction, that is, reproduction involving sex, we have a male and a female. So the male gives a gamete, the female also gives a gamete. So these two gametes mix, fuse together to form the offspring. So that means the male guinea pig has to get its own gametes. The female gets its gametes. So then the gametes will then mix together to form the offspring. So what are the possible gametes from the male? So the possible gametes are this. When it comes to the color of the fair, the male can give capital B. It can give capital B. And when it comes to the length of the fair, it can give capital S. So that is one possibility. Maybe capital B, capital S. Or it can give capital B, and then instead of giving capital S, it might pair it up with small s. That's another possibility. There is a third possibility. Instead of capital B, it might give small b to the offspring and then pair it up with capital S. Or it might give small b and pair it up with small s. So these are the four possible gamete type that the male can give or contribute when it comes to forming of the offspring. Now remember that the male genotype is the same as the female, meaning the gametes will be similar to the male's gametes. So this will be the gametes for the female. So these are the gametes that will be combined to form the genotype of the offspring. So now let's use the Punnett square to form that. But before then, take note of this. The gamete from the male fuses with the female gamete, or the gamete from the female, to form an offspring. So let's see that on our next slide. So let's assume this. So for the vertical section, those are the gametes for the male, and for the horizontal, those are the gametes for the female. So let's look at the first one. So the combination, this is a possible combination. We can have capital B, capital S, meaning the male brought its gametes, capital, those are dominant traits, and the female also brought dominant. Or the male brought dominant for both traits, and then the female brought one dominant and one recessive. And when you move to the next, it can be capital B, small b. So the male contributed capital B, and the female contributed small b. Or the male contributed capital S, and the female also contributed capital S. So with the later understanding we've gotten from the first three that we've done, can you guess what the remaining will be? Check this out and see. So it's all about pairing up. You pick one from here, then you pick, then you match them up. You pick, you match them up in that way to complete the Punnett square. If at this place there is a section you don't understand, you can just ask a question about it in the comment section. But take your time to analyze the Punnett square very well to understand what is going on. So this is what you've been asked to do. So this is a Punnett square to determine the possible genotype of the offspring. Now our next question is this: what is the phenotypic ratio of the offspring? What is the phenotypic ratio of the offspring? Let's go back to our Punnett square, and that is going to help us to tell what the phenotypic ratio should be. You see, from this Punnett square, that is 4 gametes from the male, possibly combined with 4 gametes from the female, meaning we have 16 possible genotypes that can be formed. So we are to find the phenotypic expressions of these genotypes. What I mean is the genotypes, capital B, capital S, those are the genotypes. How does it express phenotypically? Express outwardly for us to see. So let's look at the phenotypic expressions for this genotype. So before we do that, let's expand, let's widen the table. So let's pick the first genotype, capital B, capital S. Now remember, the B's are responsible for the color of the fur, and the S are responsible for the length of the fur. So capital B, that should tell us automatically that the fur color is going to be black, and then the length of the fur is going to be what? Short. So for physical expression of this genotype, it will be black fur, black short fur. Let's put it that way. A black short fur. What about the next one? What do you think it should be? Capital B. Since there is even one capital B there, it means it's going to be black. And there's one capital S over there, means it's going to be short. So that is a trick. If you see one capital B, go for black. And if you see one capital S, go for short. So let's do the next one. The next one is going to be black short guinea pig. And the fourth one is also going to be black short. So with this little understanding, what do you think the next one will be? It's also going to be black and short. But look at the next one we have. Capital B. That means the color of the fur is going to be black. What about the length? Will it be short or long? Remember, I have small s, and S is responsible for long fur. We don't have capital S over there, so it can be short. We have 2 recessive traits for the length, so that is going to be black, long fur. Can you guess what the next 2 will be? Yes, black short and black long. Now let's see if you can guess the next one. So compare our answers to what you think the answer should be. What do you think the answer of the small b, capital S will be? I think you've guessed it right. The B's are responsible for the color, right? They're responsible for the color. So if we have small b, 2 of them, those are 2 recessive traits. It basically means that the color is going to be white. So we have small b means it's going to be a white guinea pig that has a short fur. And the next one too is small b. So it's going to be white that has a short fur. Now let's predict the next three. Check them: black short, black long, and white short. Were you able to predict them? Or do you understand how we got them? Now for the last one, what do you think the answer will be? Pause the video. You can write your answer in the comment section. Are you done writing your answer in the comment section? Small b, small s, small s. Remember, the B, small letter B is responsible for white fur, and small letter S is responsible for long fur. And those are recessive traits. So if two of them are present, the recessive trait will be expressed phenotypically. So this is going to be a white, long fur rabbit. So these are the genotypes and their corresponding phenotypes. But the question is asking us to find the phenotypic ratio. So we are done with the first step. The next step is to pick each of the phenotypic expressions and count the number of times it appears. So the first one is black short guinea pig. A black short fur guinea pig. A guinea pig that has a black fur and short fur. So let's count the black short fur guinea pig. If you count them correctly, you realize that we have 9 of them in there. So the black fur guinea pig that have short fur, there are 9. Now we are done with the black short. What about black long? So now look through and count the ones that have black fur that is long. You realize that the black long ones are 3. What about a white short? Now let's go to white fur guinea pig that have their fur being long. If you count them correctly, they are also what? 3. And the last one will be a white guinea pig that has a long fur. That is only 1. A white guinea pig that has a long fur. So these are the number of offspring that are possible to be formed from this combination. So what is the phenotypic ratio? So the phenotypic ratio is 9:3:3:1. 9:3:3:1. This is what it means. If you count the number of offspring from this crossing possible, we have 9 plus 3 plus 3 plus 1. That is what? 16. That means out of these 16 offspring, possibly, this is probability. Out of these 16, there's a possibility of 9 of them having black short fur. And there's a possibility of 3 of them having black long fur. And a possibility of 3 having white long fur. So a white short fur. And then there's a probability of only 1 out of the 16 having what? White long fur. That's quite interesting. Okay. So this is a summary, a table that summarizes everything. A table that summarizes everything. If you are interested, you can take a time and zoom in and analyze what you have. This is similar to what you did on previous slides in a way. Now let's move on to our last question, final question. So what percentage of offsprings will have white long fur? What percentage of offsprings will have white long fur? Before I answer this question, let's look back to our previous slide. Remember this phenotypic ratio, 9:3:3:1. If you look at the screen, you can see that only 1 has white long fur. And we have to find the percentage of them that have white long fur. So let's get back. So you should take note that only 1 out of 16 will have white long fur. So what that means is they have 1 divided by 16, 1 out of 16, times 100. And 1 divided by 16 times 100 gives us 6.25%. So there is a 6% chance of this crossing to have white long fur, which is quite low. So you can do your own percentage for having black short fair. You can calculate it right now and put it in the comment section. That is a 9 out of 16. Calculate it, let's see. So 9 out of 16 times 100. You can use your calculator. Go there and check it out for us. So you write your answer in the comment section. You see that it is surprisingly high. So basically, this is dihybrid crossing. And we've done it using guinea pig. You can apply the same strategy with even humans. So you are observing how a certain trait, maybe the shape of the nose and then the ear, these two traits are passed to offspring. And genetic crossing becomes also important when it comes to genetic counseling before marriage. Maybe the partners involved, their sickle cell status. So if you know their genotype for the sickle cell status, then based on that, you can do your own calculation and tell the percentage or the probability of they having a sickler as a child. So this is how amazing and interesting genetic crossing can be. So this brings us to the end of our lesson on genetic crossing. If you find this video interesting, give it a thumbs up, like the video, subscribe to our channel to stay updated for our next lesson. See you next time.